Assalamualaikum warahmatullahi wabarakatuh. My name is Muhammad Afiq Arsyad bin Azahari and my metric number is 2311901. And here with me is...
Hello, my name is Iman Haziq bin Talib Sabri, metric number 2312861. In this video today, we will explain about the differences between X-ray diffraction and electron microscope.
So, without wasting any time, the agenda of this video is: the first one, we want to know what is X-ray diffraction. Secondly, what is electron microscope, EM. And the last part of this video, we will learn about the differences between them, which is X-ray diffraction and electron microscope.
Did you know X-ray diffraction uses an X-ray diffractometer, which is a scientific instrument used to analyze the crystal structure of materials. It uses X-rays to probe the arrangement of atoms in a crystal, and the resulting diffraction pattern can be used to determine the crystal structure. There are several components in an X-ray diffractometer, such as the X-ray source. The X-ray source generates a beam of X-rays through the sample. The sample is placed in the path of the X-ray beam to determine its structural composition. Lastly, the detector measures the intensity of the diffracted X-rays. Some of the well-known applications of X-ray diffractions are determining the crystal structure of materials, studying the arrangement of atoms in molecules, measuring crystalline size and strain, and identifying and quantifying phases in mixtures.
Have you heard before the word electron microscope? Actually, electron microscope is a type of microscope that use electrons to magnify and visualize objects. Electrons have much shorter wavelengths than visible light, so electron microscopes can achieve much higher resolution than light microscope. This allows scientists to see objects in much greater details, including the internal structure of cells and virus. There are several main parts in an electron microscope. The first one is electron gun. The electron gun generates a beam of electrons. Number 2, condenser lens. The condenser lens focuses the electron beam onto the sample. Number 3, sample. The sample is placed in the path of electron beam. Number 4, objective lens. The objective lens magnifies the image of the sample. And lastly, the main part is projector lens. The projector lens projects the magnified image onto a screen or camera. Generally, there are two types of electron microscope. The first one is transmission electron microscopy, TEM. Electron beam is transmitted through the sample. This is useful for studying thin samples, such as slices of cells or tissues. The other types of electron microscope is scanning electron microscopy, SEM. The electron beam is scanned across the surface of the sample. This is useful for studying the surface structure of sample, such as the surface of cells or virus. Nowadays, electron microscopy has a wide range of uses, including: the first one is studying the structure of cells and virus. Secondly, analyzing materials. The third one is studying the surface of samples. And lastly, electron microscope is used in investigating medical condition.
Now that you have known about X-ray diffraction and electron microscope, we can now compare and find the differences between XRD and EM. The interaction with matter for X-ray diffraction is the X-ray beams are diffracted by atoms into different directions, whereas for electron microscopy, the electrons are interfered by the atoms. Next, the wavelength for X-ray diffraction is higher than electron microscopy, specifically 100 picometer for X-ray diffraction and 0.1 picometer for electron microscopy. The resolution for X-ray diffraction is visibly lower than electron microscopy. On the other hand, the penetration for X-ray diffraction is higher than electron microscopy.
In addition, the cost involved in X-ray diffraction is much lower compared to electron microscopy. Besides, the advantage of using X-ray diffraction is it can penetrate thick sample. It also can be used to study non-crystalline materials. The advantages of using electron microscopy is it can provide high-resolution image of cells and tissue. The disadvantage of using X-ray diffraction is it cannot provide as high resolution as electron microscopy, while electron microscopy also cannot penetrate thick samples.
Thank you for watching. Till we meet again in the next video. Assalamualaikum warahmatullahi wabarakatuh.